One last quick thing,
Yep,
guys.
yep.
Name and location in the chat for attendance. I will call for your CEU information at the end. Take it away, Lori.
Okay, thanks, Graham. Sorry, my video is also not working for some reason, so you don't get to see my face, but that's probably okay. As long as you can see the slides. And I think Chrissy sent handouts earlier today, so hopefully you have those to refer back to as well. So today we're going to talk about tree biology, and it's like I said, can be a little tedious. For many of you, you probably know a lot of this, but some of you might be just getting into the arboriculture profession, and maybe this will be some new stuff to you. The Arborist Certification Study Guide has actually, that first chapter has 96 keywords in it. So we're not going to go over all 96 keywords. So you'll have to refer back to the chapter if you're going to be taking the exam and learn some of these words as well. But I'll try to cover several of them, and as well as talk about how they might apply in some situations. But much of what you're learning in some of the other weeks during this tree care workshop series is going to talk about the application. But it's good to have this background biology to understand how and why you're doing things when you're working with trees. So tree biology is basically the study of the structure or the anatomy of the tree and the function or the physiology behind the tree. All right, so we're gonna start off here with basic structure of trees. And the basic structure of trees and plants and organisms in general is the cell, which is basically a unit within tissues. There's typically a membrane and then a cell wall around that membrane, and then certain internal, what they call organelles within the cell that help it to function. So here are some cork cells that you might see in a piece of cork. And here is some of the chloroplasts within the leaf tissue. And so you have a leaf cell here containing these little green chloroplasts, which are little organelles that allow for photosynthesis. And cells grouped together form tissues. And these actually perform a certain function within a tree. And there are three basic tissues: meristematic tissue. We will talk about meristematic tissue in a little bit, which is basically a tissue that divides and produces other cells. Vascular tissue, vascular meaning transportation. It moves materials from one location to another. So vascular tissue is another type of tissue. And then there's epidermal tissue. And epidermal means it is an exterior type tissue. Epi means above, dermal means basically tissue or layer. So something that is externally an external material on top of the rest of the tissues below, epidermal. All right, and then if you have a group of tissues, it's called an organ. And these are familiar to all of us. The leaves are organs, stems are organs, roots are organs, flowers, fruit, these are all organs within the tree. And then if you, excuse me, have a group of organs together, it makes up the organism or the tree. So that's your basic structure. So we're going to go ahead and start with looking at some of these more specifically. And we're going to start off with leaves. So the main function of leaves is for food production, producing the energy that trees need to function. And this is accomplished with a green pigment called chlorophyll. This chlorophyll pigment captures the energy from sunlight and converts it into energy for the plant. And as I mentioned before, within the cells of the leaves, there are little organelles that are called chloroplasts, and these chloroplasts contain the chlorophyll within them. And as you can see, there's a lot of chloroplasts in these leaf cells. There's other pigments in leaves as well. Anthocyanins tend to run in the sort of the red, purple, blue hue, and anthocyanins actually have a protective function in leaves. They help to protect the chlorophyll and the leaves from excessive sunlight. It's complicated. I don't really quite understand how they work, but they do have a protective function. Carotenoids are your orange yellow pigments, and they also work in capturing light, and they can capture that energy from light and transfer it over to chlorophyll. So, and these are the colors that you often see appearing in the fall after the chlorophyll starts to break down. And once that chlorophyll breaks down, some of these other colors start to appear in the leaves. So let's look at the anatomy within a leaf. So if we were to cut a leaf across this way, this is what we would see. And this is a diagram that's in your book. And so we'll just kind of start from the top here. The cuticle. The cuticle is kind of a waxy layer that's on top of the leaf surface, which helps to prevent evaporation, loss of water from the leaf. And below that cuticle is your upper epidermis, the sort of that outer layer that I mentioned before. And then as we come down, this is called the palisade layer. Palisade, I believe, means wall. And if you look at it, they're lined up nice and neat, these cells are. And it looks like kind of a wall that you might see around a castle or something. So that's your palisade layer of cells. And then down in here is called a spongy layer. And it doesn't really show real well in this diagram, but the spongy layer is cells that are sort of more loosely combined in the area with a lot of air space around them. And it is these two layers where you have a lot of that chlorophyll present. And this is where that photosynthesis is occurring that captures that energy from the sun. And then we have, this is representing the leaf vein. The vein is your vascular tissue, your conducting tissue. This is where the xylem and phloem occur. And we'll talk about xylem and phloem a little bit later. Here's your lower epidermis. And then we have what are called the stomates, which are usually on the lower surface. These are openings into the leaf that allow for air exchange. They're called stomates or stomata. A singular is stoma. And surrounding these openings, these pores, are what are called guard cells. And these guard cells regulate the opening and closing of the stomate. So that's why they call them guard cells. So this is the diagram that's in your book. It's pretty basic diagram. This is a little bit nicer diagram that I thought I'd show you. Again, showing the same layers, the cuticle, your upper epidermis. Here you can see your palisade layer of cells. And this shows a little better the spongy layer with all the openings in here, which allows for air exchange, gas exchange. And then of course your guard cells and stomates in your vein in here. All right, so looking at the bottom surface of a leaf, you can see under magnification the guard cells and the stomates or stomata. Sorry, that's all covered up back there. It just said stomates or stomata. And it is through these guard cells that you have gas exchange occurring. Carbon dioxide from the atmosphere is going to move into these openings and is utilized in photosynthesis, which we'll talk about in a minute. And everyone, you know, kids all learn that green plants are good because they produce oxygen. Oxygen is a byproduct of photosynthesis. So oxygen is released through these guard cells, through these stomates. And then the other thing that's happening is water is also lost through these stomates. So the water that gets picked up from the soil, through the roots, through the stems, all the way up through the leaves, eventually makes its way out through these stomates. Now, the loss of water through the stomates has an actual term, and this is a key term, transpiration. So loss of water from the leaves is termed transpiration. All right, let's look at photosynthesis. You can think of photosynthesis as an energy transfer system. Energy transfer system. So the green leaves capture energy from the sun and convert it into plant energy so that the plant can utilize that energy to do things. So an energy transfer system. You can also think of photosynthesis as a carbon transfer system. So the carbon dioxide in the atmosphere is captured by the green leaves, and it is converted into carbohydrates. So you have carbon and then carbon. And it's in those carbon bonds in the carbohydrates where the energy is actually present. So it captures that energy and puts it into carbohydrates. So I'm going to mention carbohydrates, carbs, sugar, starch, energy, all kind of as the same thing. So that I will be mentioning that throughout the talk this afternoon. Let's see. So photosynthesis. Here's kind of the reaction of photosynthesis. You have carbon dioxide, you add some water, and then capture that sunlight energy and you convert it into carbohydrates. And as a byproduct, you have oxygen. So carbon and water hydrate carbohydrate. That's your photosynthesis reaction. And what you're getting out of this, carbs, is energy for the plant. So carbs are good. We kind of think of carbs as, you know, maybe not being good because we're all on, you know, low carb diets, you know. Well, carbs are good. Sugar is good for the plant. It means energy for the plant. So carbohydrates are used for several things within the plant. Maintenance, just general staying alive, maintenance, but also growth. So what trees will utilize a lot of carbohydrates just to grow. Reproduction, producing seeds, fruit, flowers, all of that uses carbohydrates, energy. They will put some of that carbohydrate into storage so that it can be used in the future for needs in the future. And of course, there's defensive compounds that are used, that are made with carbohydrates. So there's a lot of uses for this energy that is captured from the sun. All right, a couple more terms that are in your book. source and sink. A source is a plant part that gives carbohydrates to another plant part. And the sink is the plant part that receives those carbohydrates. So here's a little picture of that you can use in your mind to remember source and sink. Your source would be like your faucet. Your sink, of course, is the plant part that receives the carbohydrates, and the water here is representing carbohydrates. So source and sink, source and sink. So here are some examples of sources and sinks. Your source, leaves are typically going to be your source. They are giving carbohydrates to another plant part. So in a fruit tree, the leaves that are, in particular the leaves closest to the fruit, are the ones that are providing the carbohydrates to the developing fruit, the sink. So there's your source-sink relationship there. Another source-sink relationship would be the leaves sending carbohydrates down into storage in the trunk or in the roots. So the leaves are the source, the trunk or the root system becomes the sink because they receive those carbohydrates. Now, sometimes leaves can be a sink. When they're very, very young, the leaves are going to need carbohydrates to develop. And if you take a look at the interior part of a twig and you stain, this is a twig that's been split open and stained with potassium iodide. And if anybody has ever done this as a high school experiment, you can take this iodine stain and place it on tissues. And if there is starch present in those tissues, it will stain that starch a real dark color. And so what you see here in this twig are these dark areas right underneath the buds. There is a lot of starch stored in here in these twigs. And in fact, there's a lot of starch even in the buds themselves because the buds are very dark too. So in this case, we have a source being the twig, and the new developing leaves become the sink because they are growing, and so they are needing a lot of carbohydrates to grow. So going back to our photosynthesis equation here, carbon dioxide plus water plus energy or sunlight becomes sugars and oxygen. Now, the reverse of photosynthesis is respiration. So when the plants need to actually use the carbohydrates and get the energy out of the carbohydrates to do something, they have to go through the process of respiration. And that's just basically the reverse of photosynthesis. You take the carbohydrates, you add some oxygen, and then you release carbon dioxide and water, and you get that energy which the plant can use to do something, to grow, to produce fruit or seeds, or to just maintain the plant's existence. All right. Now, this is a couple of terms that confuse people sometimes, so I thought I'd point this out. The difference between respiration and transpiration. So going back one here, respiration was sort of the reverse of photosynthesis. It requires oxygen. Respirations, we talk about respiratory diseases. That means diseases of our lungs. Okay, respiration requires oxygen, and that oxygen is what is used to break down the carbohydrates into usable energy. So respiration is breaking down of those carbohydrates using oxygen. Transpiration, if you remember just a few minutes back, we talked about transpiration. that is the loss of water from the leaves. So keep those two terms in mind when you're, especially if you're working on that exam. Okay, let me pause here a little bit before I go to meristems, and if anyone's got any questions, go ahead and ask them now. Is there anything in the chat box, Graham?
No, haven't seen anything in the chat yet, but feel free to put any questions in there, or if you wanted to, you know, right now while we're taking a quick break, if you wanted to unmute yourself and ask a question, you could do that too.
Yeah, I'll just wait for just a few seconds.
Yep, nothing in the chat.
Nothing. And that's fine. We'll have time, I think, at the end to take questions as well.
You bet.
All right, let's go ahead then and move on to meristems. Meristems. So what is a meristem? It says meristem at the top of my slide here. This is a tissue that has actively dividing undifferentiated cells, and it produces other tissues. Undifferentiated cells. That means these cells are just a basic cell. There's nothing special about these cells. They don't really have any sort of function other than to divide. They divide, divide, divide. That's what meristem cells are. You could kind of think of meristem cells as stem cells in humans. They're just your basic cell that can divide and eventually produce any other kind of cell. So meristems. Now in plants, we have a couple different kinds of meristems, meristematic tissues. There's your primary or apical meristem, and this produces growth in length. Okay, so here's a little motion here. Growth in length, primary or apical meristems. And these are found in the buds or the shoot tips and in the root tips. So here's a little picture here of a shoot, and you've got a primary or apical meristem in the bud at the very tip. So here is a magnified view of a bud, and this tip is where all these cells are actively dividing, and they are producing more cells that eventually differentiate and become other kinds of tissues within the stem. But that's your apical meristem in your shoot. And then at the root tips, of course, you have an apical meristem as well. Again, actively dividing cells that eventually will produce your root tissues. And on a root, you have, because the roots are in soil, that apical meristem would be damaged if it was just out there exposed to the soil. So there's always a little root cap, what they call a root cap, on the end of the root tips. All right, so Graham, this is where I would show that first video.
Yes.
So if you're able, do I need to stop sharing here?
You can, yeah.
Okay.
And
So
we are, yeah.
Yeah. I've got a little video that you can watch. And Graham will show that here.
Yeah, yep, I'm getting there.
All right. This is just a simple video of a bean. It's a bean seedling growing. And it's going to show you your apical meristems and how they are growing in the soil and above soil. So I don't hear the sound. It's just music, not a big deal. But you can see apical meristems at the tips of each of these roots. And then, of course, your apical meristem at the shoot tip is going to produce your shoot. And what you'll notice too is that, you know, the shoot after the apical meristem produces the cells, there's a point in the stem where you have elongation of the cells. And then after those cells have elongated, nothing else is going to move upward. So the cotyledons here stay at this height. This first set of leaves will stay at that height. There's no movement upward of these leaves because your apical meristems are at the top, at the point. Okay. And you see a lot of motion in here. This is a bean plant, and beans, they like to twine around something. So I don't understand quite how this movement is occurring in this. It might be certain hormones or something, but the beans are searching for something to twine around is what's going on here. And if they were to find something, you know, a fence or something, then that stem would start wrapping around that fence. Okay, so we're back to the roots again. We can stop here, Graham.
Very good.
Okay.
No, that was interesting. You know, I'm sure that there's some phototropism going on with the twisting around and around, and then some other kind of response when it actually encounters a physical object that it can continue to twine around.
Yeah, and that's another term, the phototropism. So a tropism is a plant movement that responds to an external stimuli. So a phototropism would be a plant responding to light. And so a plant might grow towards light, and it's turning towards the light because that's called a phototropism. You also have geotropisms, and that's the direction that a plant moves in response to gravity. Geo, ground, so gravity. Tropisms are a couple other terms that are in your book that you should review. Okay, let's go back to, oh.
Yeah, there you go.
Okay, so we'll go back here and share that. So, all right, let's go back down to the next. Okay, all right, so we're back to, that says meristem up there. So we talked about the apical meristem. There's also a secondary or lateral meristem. And this is a meristem that produces growth in diameter or growth in width. And so there are a couple of lateral meristems or secondary meristems that we're familiar with. The cambium or the vascular cambium, that's the one most of us are familiar with, which we'll talk about quite a bit more here in a minute. But there's also a cork cambium, and this cork cambium is found in the bark layer, and it actually also produces some width, some diameter growth within the bark. So there's the cambium, and then there's the cork cambium. So looking at cambium, the cambium meristem, the cambium layer is actually a very, very thin layer of cells within the stem of trees. And it produces cells to the outside and to the inside of the cambium layer. And the cells that are produced to the outside of the layer become phloem. They differentiate and become phloem. And the ones that are produced to the inside differentiate and become the xylem tissues. And then of course you have your bark layer out here. And the bark also has a cambium layer, the cork cambium. And I'm not really going to talk too much about cork cambium other than to mention that there is a cambium in that bark, which is producing some layers of tissues in the bark as well. Okay, so just a little question in your mind, and you should all now be able to answer this. Where will the side branch be in 3 years? So on a tree, of course, you're not going to have those branches once they're produced. They're not going to be moving up the stem. So this is going to remain at the same height year after year after year. And in fact, many of these branches are going to ultimately probably have to be removed because they're just going to, you got to have some clearance here as you're, mowing under the tree. So most of these branches ultimately probably will get removed, and your main scaffold branches will probably be up here somewhere eventually. All right. Any questions at this point before I move on to stems?
I'm not seeing anything in the chat.
Okay.
We're doing good.
All right. I think we'll keep going. I don't have my little clock here to see how I'm doing time-wise. So, all right. Let's look at stems. So if we look at twigs, just looking at the parts of a twig, that terminal end bud is known as the terminal bud or the apical bud, that last bud at the end of the twig. And then you have on the sides, lower down, what are called axillary or lateral buds. And in the spring, when we have bud break, these buds are going to produce your new shoots, your terminal shoot and some side branches. And ultimately, these then become your new twigs in the future. So another aspect to look at on twigs are what is called bud scale scars. And that's this horizontal line here, which encircles the twig. The bud scale scar is produced when the scales from the bud, which surround the bud, fall away in the spring and leave behind a little scar. So last year, there was a terminal bud right here. And then when bud break occurred, those scales around the bud fell away where they were attached, and they left behind the little bud scale scar. And then the twig grew out just a short amount, and then it set a new bud up here. So you can actually look at a twig and look for the bud scale scars and get some idea about the growth that has occurred year after year after year on the twig. So looking at this twig, you can actually determine how many years of growth is on a twig. And if you take a look carefully for the bud scale scars, there, it's these horizontal lines that would encircle the twig. You can see that this twig has 1 year of growth, 2 year, 3 year, 4 year, 5 years. This twig is 5 years old. So the terminal bud was here in the spring. The bud scales fell away and it grew out this much and set a bud there. And then the next year, the bud scales fell away and it grew out this much and it set its terminal bud right there. So each year it just grew out just a very short amount here. And when I give this talk to like the master gardener groups, when we did this before COVID, I was able to bring twigs to the presentation and distribute them among the master gardeners and let them examine actual twigs so that they could practice looking for bud scale scars, which is a good thing to learn how to do because it can give you some idea of how much growth is occurring on the twigs. And so it can give you some idea of the general health of that tree. So this twig is showing, you know, 5 years of growth on just one short twig. Now, if we look at this twig over here, how many years of growth do we have on this twig? And I usually have all the master gardeners raise their hand when I say, is there 1 year's worth of growth or 2 years worth of growth? And in this case, there's only 1 year's worth of growth on this twig. What we're seeing here, these two buds here correspond to these two buds up here. There are no bud scale scars on this entire twig. There's no horizontal lines to indicate a bud scale scar. So this is all 1 year's worth of growth on this twig, which indicates that this twig is growing well. It's produced a good length of twig growth compared to this twig over here. And then I mentioned that the environment these two trees were growing in is quite different. So this slow growing twig came from a tree growing in brome grass, which is a very competitive grass. And so the tree was under much more stress than the tree growing in a wood chip mulch, where it was able to grow quite well with a good, healthy root system.
Lori, there was.
Yes.
Sorry to interrupt you.
Nope.
There was one question in the chat about distinguishing between stem buds and the ones that will produce leaves. Okay.
Let's go back. We'll go back to this picture here. So these buds, when they open up, they are actually producing the stem tissue, and on that stem tissue, you have your leaves. So you've got a shoot coming out, and that shoot is going to become your stem, your twig, and it's going to have those leaves attached to it. The only other thing that might be going on is that sometimes you have buds on some trees that have both shoot tissue, stem tissue, and flower tissue in there together. So you might have a bud that has both, that will produce both stems and flowers, or sometimes you have buds that are separated. You have a floral bud that produces only a flower, while you have a vegetative bud that produces only your stem tissue.
Okay, so that means that every bud is going to have a shoot and foliage coming off of it in whatever organization that species tends to do.
Right, correct.
It's just a question of whether that's a terminal bud or, you know, a subordinated kind of lateral.
Yeah, right. So you have your terminal bud producing your terminal shoot here, your main leader, if you will. Right. And then your axillary buds are producing your side shoots or side branches.
Great,
so
thank
hopefully,
you.
Yeah, hopefully that answered the question. All right, so let's look at this picture again. You'll notice on this slow growing twig that all the buds are at the very tippy top of the twig, while on this good growing twig, the buds are going to be spread apart. So when you've got your new shoots coming out, this twig is going to be nicely fully leafed out, while on this one, the leaves and stems that come out here are going to be near the top of the very ends of the twig. So when you see branches that are growing slowly, you'll notice how the leaf tissue tends to be located at the very ends of those twigs. So when you see a thin crown like this, it's probably because all those buds are at the very tippy top of those twigs. Here's just another picture of a twig, and can't quite see the bud scale scars here, but there's one here. And then you got some really good growth that year. Then it kind of slowed down, not quite as good a growth that year. And then again, very slow growth. So again, looking at a twig, you can kind of see what kind of growth it's been showing over the years. And you can see that maybe something happened just a few years ago that started to affect the health of the tree and slow down its growth. Now you have to also kind of look at some trees and see, are they putting on a second flush of growth? So it's not necessarily going to be 1 year's worth of growth. In this case, here's an oak, which again, it's kind of hard to see in this picture, but you had the start of growth in the spring, which ended here. And you can see these leaves are older. And then the same year, it put on a second flush of growth, which was really good. The conditions happened to be really good for more growth. So you got a second flush of leaves later in the season. And so you would have a bud scale scar there and a bud scale scar here. And this actually all occurred in 1 year. So, you know, there's never anything absolute in nature. There's always exceptions. There's always gradations. So you kind of have to sometimes look at things as just a general rule as opposed to an absolute rule. Again, here's another thing that might trip you up: epicormic shoots. So these stems popping up from the trunk here represent all 1 year's worth of growth. Okay, long stems popping out from the trunk. There's not going to be any bud scale scars along this entire length. So it might make you think, oh, good growth is occurring here. This tree must be healthy. But actually, when you have epicormic shoots, it's an indication that the tree is actually under some stress. In this case, in this picture, this is an ash tree that is being attacked by emerald ash borer. And you can see how very thin the foliage looks up here. And what happens when you have epicormic shoots is that you usually have some kind of injury to the branch above, which is killing off the branch above, and it's stimulating the production of these epicormic shoots. Epicormic shoots, epicormic sprouts, water sprouts, suckers, those are all kind of the same thing. Those are all the same thing. And you can tell an epicormic shoot from just a normal shoot because they are small diameter attached to a very large diameter trunk or branch. So very, very small diameter twigs attached to a very large diameter trunk usually means it's an epicormic shoot or an epicormic sprout. And they usually are quite long and grow a great deal in one season. And these epicormic shoots come from dormant buds. Dormant buds is what it says under there. And these there's other terms for them. The book uses different terminology. Adventitious buds, dormant growth points. These are all the same thing. These are what these are little growth points right underneath the bark, and they're dormant. They don't do anything unless they are stimulated to grow because of some type of injury to the branch above it. And if there's damage, let's say a bad pruning cut, looks like we got a flush cut here, and you can see we've got some damage down here on the branch. This damage here may have affected the hormones, the plant hormones, such that it allowed these dormant points underneath the bark to become activated and produce these epicormic shoots. Okay, it's kind of a backup system for the tree to produce some new growth when it gets injured. But it's not a very good condition. It usually, when you see epicormic shoots, it typically means you've got some serious problems in the tree or some injury to the tree. And some of that injury could be topping. Topping is a serious problem in trees. It's a bad practice. And what it tends to do is initiate or trigger epicormic shoots in a tree. And topping is when you're cutting through a branch right in the middle of the branch. And that injury stimulates those dormant points to grow, and you get these epicormic shoots that develop. In this case, it was cut. The cut was made right across the middle of the branch there. It stimulated these sprouts to start growing. You'll notice too that the main branch started to decay out. And here's kind of a cut through that branch that shows how much decay has occurred, and these epicormic shoots up here, and how very little live tissue they are attached to the main branch. So as these shoots get bigger and bigger year after year, they start to put on some weight, and they're just not very strongly attached to the main branch. And so that means they're more likely to fail under bad conditions. So here's a couple of pictures that one of our prior foresters, Chip Murrow, took, which I use a lot. This is a tree that was topped, and then it produced all these epicormic shoots in response to that topping. And then there was an ice storm which put a lot of weight on those epicormic shoots, and they all failed because they just don't have that strong connection to the main branch. Here's another term, included bark. Included bark. So included bark is bark that has been trapped in between two stems on a tree. And you often see included bark on stems that are growing in a very tight V-shaped crotch. Notice how close together and vertical these two stems are. And as they grow in diameter, the bark tends to get pinched in between the two stems. And so you end up with bark against bark internally in here. And you'll usually see a seam running down between the two stems when you have included bark. And because you have bark against bark, instead of a nice strong connection of wood, there's a likelihood that one of these stems could split out from the other. And here's a couple of pictures that show the included bark in a branch that failed. So this area right in here, and here's the tree, the branch that fell out. This area right in here is the included bark, the bark that was in between those two branches. And you'll notice this is the wood connecting the two pieces together. And there's just not a lot of wood holding those two pieces to each other. Most of it was blocked by this included bark in here. So this is something that should be watched for in young trees and eliminated with pruning while the trees are still young. Another term is the branch bark ridge. And these terms are going to be talked about probably more when we go over pruning in future sessions. but just to introduce the biology behind it, the branch bark ridge is the bark that sort of gets pushed up between the bark of the branch and the bark of the trunk. And you see branch bark ridges in wide angles as opposed to these narrow V-shaped angles. When you have a wide angle, you don't get that included bark trapped in between those two. Instead, the bark is getting pushed up and out into a ridge, and the connection between these two, the trunk and the branch, is wood. You got wood interlacing in there. So you have a good, strong branch connection. So you look for the branch bark ridge, and you also look for what is called the branch collar. Sorry, that's covered up there. Branch collar. And the collar is simply a combination of branch wood and trunk wood that is wrapping around the branch. And that wood in there is actually kind of layered over each other year after year so that you have a nice, strong connection. And a branch collar is often kind of a swollen area that is wrapping around the branch. And you can kind of see these branch collars here. And in fact, in this last picture, you can actually see kind of a swollen area right in here, which indicates the branch collar as well. And the branch collar is important to identify if you can when you're pruning, because you don't want to damage the branch collar when you prune off a branch. You want to stay outside of that branch collar, and you would make your cut out here. If you do a flush cut through the branch collar, you are actually damaging trunk tissue, and you're setting up the tree for problems in sealing off that wound and protecting the trunk. And sorry, this is covered up, but you have a branch protection zone within the collar. And this branch protection zone in this collar area and in this collar area is an area which has properties that help to protect the trunk from invasion by fungi, decay fungi. So if you were to do a flush cut straight up and down to cut that branch off, you would be cutting and removing that protection zone. And then you'd have this big open wound exposing the trunk to decay fungi. If you make your cut properly outside of the branch collar, you maintain the integrity of this protection zone, which can then better fight any fungi that might be trying to enter through that pruning cut. So you don't want to flush cut. You also do not want to leave stubs. So you don't want to cut way out here either, because you end up with a branch piece that starts to decay, and that decay fungus is able to live off the food of the stub and become more strengthened and potentially may be able to overcome this protection zone and enter into the trunk. So you don't want to leave a big food base by leaving a stub. And also those stubs mean that that wound is not going to be able to close over. It would just take forever for this tree to eventually produce enough closure until it reaches this end. So don't flush cut and don't leave stubs. And the pruning will be talked about in the future. I don't have a lot of experience with pruning, so this is about as far as I can go in explaining pruning. So I think Graham will probably be the one that will talk more about pruning. So just showing a proper pruning cut outside of the branch collar. A good pruning cut is going to produce wound wood in the shape of a donut. so that it will seal very quickly. If it's been cut too close, you damage that collar and you don't get a nice donut forming around that pruning cut. Now, just real quick, the difference between the terms wound wood and callus. Some people like to say a pruning cut will callus over, and they call this callus. That is incorrect. Callus is undifferentiated cells. It's kind of like meristem cells. It's just a mass of basic cells that have no specialized functions. They have no specialized structures. They just divide. And whenever you make a cut, a tree will respond by producing callus, but the callus is just this fragile, simple cells that divide. Eventually, you start to get differentiation, and the cells will start to differentiate. They'll start to develop certain features about them. They might get some lignin incorporated into them. They become more organized, and then you have this production of wound wood. And that is what we're seeing here, wound wood. Wound wood is basically like wood, xylem tissue. And that is what's covering over your pruning cuts, is wound wood, not callus. So that's just some terminology that people get confused with. Okay, phloem and xylem. We're gonna move on, but are there any questions at this point?
I do not see any in the chat, but feel free to chime in here quick if you'd like, if you had anything you wanted to ask on that topic. Otherwise, I think we're good to go, Lori.
All right. What's our time, Graham?
Sorry, we are at 3:05, so you have about 25 minutes.
Oh, okay. I better move along quickly here. All right. So going back to cambium, phloem, bark, xylem, we're going to take a look at the phloem first. Okay, this is covered up. It says phloem, and the function of phloem is the movement of carbohydrates or sugars, and also the movement of hormones. And that phloem tissue is right underneath the bark. This would be the cambium area right in here, and then all of this would be considered xylem. And over here we have phloem tissue right in here. It's underneath the bark, and here all of this would be your xylem tissue. So the basic function of phloem, movement of the carbohydrates, the sugars. Phloem consists of living cells. Okay, phloem is alive. And these are just some diagrams of phloem. Phloem is basically tubes. Okay, the cells are stacked up in long tubes that go up and down the stem. There's certain terms for those cells in broadleaf trees, sieve tube elements, companion cells. These are in your book. I highly doubt, though, that they would ask this on the test. Conifers have sieve cells, but basically that is what's carrying your sugars in your tree. Now the bark, the outer bark, consists of old phloem and corky cells. So the phloem, there's only a small layer of phloem. And then after a while, that phloem dies away and it starts to get crushed into the layers of the bark. So old phloem can be found in the bark layers, along with some cork cells. Cork cambium is in here as well. The bark also provides defense against injury. It helps moderate temperature and it reduces water loss. So those are some functions of the bark. Now, young bark will often contain chlorophyll. And so that means it can photosynthesize. It can actually produce its own sugars. And the way that it gets the air, the carbon dioxide it needs to carry out the photosynthesis is that the bark will have what are called lenticels. And these lenticels here are just little openings in the bark tissue that allow for that gas exchange to occur. So the carbon dioxide can get into the bark layers and into this area where the chlorophyll exists. So photosynthesis can be carried out. So that's your lenticels. Okay, xylem. Xylem is your wood tissue, and this consists of dead and living cells. So the xylem has both dead and living cells. Some of the living cells, some of the dead cells need to be dead to function, while some of the living cells, of course, they need to be alive to function. And of course, we have sapwood xylem and heartwood xylem. The functions of xylem: conduction, i.e. the movement of materials, is a big function of xylem. Support, just structural support of the tree. Storage of mainly carbohydrates, sugars, and there's a defense purpose as well to xylem. So looking at a cross section, your sapwood, which contains both dead and living cells, is the outer part here. And then eventually as cells start to die and lose their function, they become discolored and you have what is called the heartwood. And the heartwood has no living cells at all. Everything has died by the time it becomes heartwood. So most of the function of xylem occurs in the sapwood. That's where the water conduction occurs. There's strength, there's food storage. The food storage is stored in the living cells in here. The heartwood, mainly, it's non-conducting. It doesn't store any food. It mainly is providing strength to the tree. So the water conducting system moving from the roots up the stem and out through the leaves. And so that is what we're talking about here. Is the movement of the water and the nutrients through the xylem in the stem. Now, water movement in conifers takes place in cells that are called tracheids. Tracheids are very long, narrow, very tiny cells, and they're stacked end to end and allow the movement of water up through these cells. They're dead when they are functional. So here's a cross section of a spruce tree that's showing tracheids. And so there's an opening, which is, these are dead cells. And so they are open and the water can flow through these cells. And you'll notice that the cell walls are quite thick on tracheids. And that's in conifers. Now in broadleaf trees, water moves through two types of cells, tracheids and vessel elements. Vessel elements are much wider than tracheids. And you can actually see in cross-section the vessel elements or the vessels. These are the pores that you would see in the cross-section of a broadleaf tree. The tracheids are so tiny that you can't even see their openings, but you can see vessels. Let's see, is there anything else to say about that? I don't think so. They have, this is some terminology from the book. You have ring porous trees. Ring porous trees are those which produce very clearly defined rings in the trees. Early in the season, you have very large openings, large pores, large vessels that are produced when the conditions are good in the spring. And then later in the spring, later in the season, when the conditions get to be a little more harsher, you have this late wood production of xylem, and the pores get smaller and smaller. And this is considered an annual ring, your combination of your early wood and your late wood. And you can often count the number of rings, of course, to find the age of your tree. Some examples of ring porous trees are oak, ash, and elm. This is just showing what are called rays, and the rays provide lateral movement. They can move from the phloem area all the way through that xylem tissue. And so they can carry materials laterally within the stem. Then you have something called diffuse porous trees. And that means those vessels, rather than being in nice, neat rings, are kind of scattered throughout the ring. And you can kind of see them. They're a little hard to see, but you can see those pores kind of scattered throughout the xylem tissue. And a lot of other trees are considered diffuse porous trees. Okay, and then just a little memory device that you can use when it comes to xylem and phloem and transport tissues. Phloem, food. Phloem, food. Phloem is what's moving your food, your sugars, through the tree, whereas your xylem moves water. WXYZ xylem. WXYZ xylem. So if that helps you to remember which tissue carries what, that's good. Phloem, food, water, xylem. Yes, question.
Lori, well, not a question, but to add to that, I also like to use the sort of word flow that's in phloem to remind me that food is moving down, like for the direction that it moves through the tissue.
Okay.
Sometimes that gets you confused with water because water flows down, but that works for me, so I figured I'd share it.
Okay, sounds good. Anything that helps you remember things. Okay, another function of xylem is support. Thick-walled cells. As I mentioned, the tracheids have very thick walls, but there's also fibers, which have extremely thick walls. You can see the tiny little central portion of this cell, but all of this is the wall of the cell in fibers. So fibers are very, very strong, very thick, and provide a lot of support. A conifer is considered a softwood because there are no fibers in conifers. Broadleaf trees are considered hardwoods because they contain fibers. So just a little note there. I'm going to keep moving here because we've got a little more to cover. But xylem also has food storage as a function. As I mentioned, the potassium iodide stain can stain starch black. And so these little areas in the xylem have been stained with potassium iodide and they've turned black. And that's because there's a lot of starch being stored in these cells in the xylem. Here's your phloem. Phloem will transport sugar, but the sugar gets converted to starch and it is stored. Starch is the storage material for those carbohydrates. So there's no storage occurring in the phloem, it's occurring in the xylem. Okay. And then another function of xylem is defense. So the xylem will produce antimicrobial compounds, insect feeding deterrents, various types of compounds, tannins, lignins, suberin, resins. Different materials are produced in the xylem for defensive purposes. Okay, now let's talk a little bit about CODIT. This is a system of defense for stems and roots, and CODIT simply stands for compartmentalization of decay in trees. So this is a term you should know. I want to get through this because this is kind of interesting. Compartmentalization. To understand CODIT, I want to talk about the Titanic. The Titanic, of course, was a ship that struck an iceberg on its maiden voyage back in the early 1900s and drowned. And there were a lot of people that lost their lives on this ship. But it does provide a good example of what's going on with CODIT. In ships, you have compartments. below the deck. And these compartments help to prevent movement of water from compartment to compartment if there is a hull breach, because there are walls here. And trees also have compartments. You can kind of even look at the cross section here and you can see these little compartments in the wood of trees. And in order for the crew to work in these compartments below ship, there are doorways between the walls, so they can move between walls, between rooms through the doorways. But those doorways are open and they could potentially let that water move from compartment to compartment. So if there is a hull breach, those doorways get closed down. And the same thing is kind of happening in trees. You have a reaction to a hull breach, if you will. You have openings between the cells get closed down. The vessels start to plug up with materials, and you get antimicrobial compounds being formed. And these are all to help prevent the spread of decay. And these walls that are within trees, are basically reactions occurring to help prevent the spread of decay. So you have 4 walls with the Codet model. Wall 1 is your upper and lower walls, and this tends to be kind of the weaker walls. Wall 2 would be the walls that prevent movement of the decay to the interior part of the tree. Wall 3 prevents the movement of the decay around the tree. And then your wall 4, which is known as a barrier zone, resists the spread into the new wood that's produced later. And it's a very chemically strong barrier. So for example, here's your wall 4. The decay has already killed off all the interior tissue, but this wall 4 is preventing the decay from moving into the new wood. So that barrier zone, that wall 4 is a good strong wall. The problem is that when you wall off injuries, every time you get an injury, you have to wall off that material and you lose energy, and this weakens trees. Plus, if you start drilling holes, for example, for injection treatments, you have the potential of breaking through that strong wall 4 and then allowing the fungus that might be inside to spread into that new wood. So while we're doing a lot of injections these days, we have to be careful about how deep we are drilling those holes, because you're potentially breaking through what the tree uses to protect itself. So breaking walls basically can sink your ship, if you will. All right. I think I'll have to stop here because of time at this point. Sorry about that, folks. We do have the handout available that you can refer to. I did talk about roots a fair amount, mycorrhizae and things like that last week, if you were here last week. So hopefully some of that would be review anyway. But I think maybe at this point it would be good to just take questions. If anyone has any questions, because it looks like we are at 3:30 at this point.
Yeah, if anybody has a question, feel free to chime in. I know those of you going after the NAA or ISA exam are probably reviewing those chapter questions beforehand. But please, if you got any of those that needed to be elaborated on or something outside of the chapter questions, we could answer that real quick here. Looks like Michael's ready to go back at snow.
Yeah. And I am going to be giving this talk again at 7. I don't know if people are, or at 5:30, right? Yep. So I don't know if people are able to get back on
Yeah.
at all, or if they only had one choice?
Yeah, that is worth mentioning. So
Yeah.
guys, get your CEU numbers in the chat for me here. But yeah, that's worth mentioning. That you can attend both sessions. You can only get CEUs for one of them. But if you just want to hear the material a second time, you can do that. And of course, as I mentioned, there's the resource page with recordings that you can refer back to on your own schedule. So all of that is available to you guys through the end of March. Yep. Thanks, Michael. Good to see you. Everybody else, get your CEU information in the chat for me. And I think that's what we got for today. I'll be covering pruning on Thursday, so I'll see you then. I love talking about this topic. It's going to be hard not to do a lot of conversation about it and just get through the concepts, just like Lori, the challenge she had today. So thanks very much. I'll leave the Zoom here open for a little bit so you guys can get your information into the chat window. So thanks so much and be safe out there.
Yeah. Thank you, everybody. I'll hang around here a little bit
Yeah. Good job, Lori.
for any last minute comments or questions.
Hey, Graham,
Yeah.
this is Phil Schmidt from Dewey. Last week, I sent you a little text or email about you're talking about getting a bucket truck and a power line.
Oh, yeah, the step potential. Yeah.
Potential.
Did you want to weigh in on that here a second?
Well, I'll just talk to you real quick. Those people that are listening, it's fine.
Yeah,
I was
sure.
an electrician for a number of years, power line electrician. And the one thing that the people that need to realize when they get in contact with the power line, that power line, and I can go up off video, that power line is energized and it will keep pushing power out there until it either blows a fuse or a breaker or something downstream or upstream. And so that truck is continually power energized. So when they jump off that truck, like you said, shuffle away from it, but they don't know how far out that's going to go. Well, with lightning, once it's discharged, it's gone. So you could walk away from the tree.
Yeah, that's a very good point. And there's kind of a I don't know how helpful this is, but there's a corollary with wind there where, down in Florida, they may get hurricane winds that are twice as strong, but don't last very long, where we get these slower, persistent winds that go on for a long time. So yeah, I really appreciate you pointing out the safety of, the hopping is not the way any longer. You shuffle.
You shuffle and you keep both your feet as close together as possible on the same plane.
Yeah.
One thing I would teach this in my safety training. Have you been down to the train station there at the depot in Lincoln where they have the water fountain?
Yes.
Okay, that water fountain's coming down and dropping into that pool. And you hear the water and there's the waves flowing away from it.
Mm-hmm.
Think of that as you're standing in that center part of that, and those waves are bouncing away from that spot where it's dropping in the pool.
Yeah.
If you touch one wave with one foot and one wave with the other, there's a potential there.
Right.
And that's where you can get current flow. So you have to be standing on the same wave. Well, as you get further away from where it's being dispersed out, the waves are further apart. So you're not.
Yep.
So I kind of use that as.
That makes a lot of sense. We're big bags of water that electricity really easily moves through, and so.
Well, no, it wasn't so much that, man, as the different waves. It's standing on two different waves at the same time.
Yeah, I get that too.
That difference in potential. One wave's one voltage, and another wave's a voltage. And when you're on a different potential there, it carries current, and that's what's carrying across your body. So you need to keep your feet on the same wave as you're getting away from the source.
Right. Yeah. Don't be in a hurry. Just.
Don't be in a hurry.
Yeah.
That's all I wanted was to make sure until you're told by somebody if your power line falls on your truck or you get tangled up with the power line, you got to consider you're energized or that ground around you is energized until somebody tells you from the power company tells you different.
Well, and I recently learned that even climbing line can easily be energized by contact with a power line. And that doesn't seem like a very conductive material, but nonetheless, people have been shocked by electrical lines through their climbing line.
Well, and that, yeah, it's, you don't know the circumstances and the voltage of that line that you're coming in contact with. And
Yeah.
you don't know how long it could be energized. There's a lot to get tangled up with power lines.
Yeah. And that's why I wanted to say up front that I'm certainly no expert. And I really appreciate you sharing your expertise there.
All right, Graham, just wanted to, and you did a good job bringing that up last week.
Well, thank you. Yeah, I'm not perfect at all, but I appreciate you, filling me in a bit more on all that.
Not a problem, sir.
Yeah, that's interesting. Thank you.
Yeah.
Yeah.
Hey, Graham, quick question too on the branch collar. There at our trailhead at Blue Springs there, people previously before did some cutting and they didn't cut, outside that branch collar. So we've got some stubs sticking out. And that's probably been about 5-6 years ago. Should we go back in and start cutting those off, those stubs, or is that going to damage more, or what do we ought to do there?
Yeah, I mean,
And they're on some nice oak. They're on some really nice oak trees.
Having the stubs at least gives you the opportunity to address the problem. You can't put it back on once you cut it off, right? If we cut too flush. So
right.
But I would base that decision on what kind of wound response you're getting from it as it is. Stubs are typically not going to seal over very well, and we can talk about this a bit more on Thursday. But if you found that the response to those injuries was pretty good, then you might want to leave well enough alone, but it's probably going to be better to get those cut back to the collar.
Okay, yeah, because they're about 2 inches, 3, but they're looking still healthy after all this time. But
the other challenge with those stubs left behind is, as Lori mentioned, there could be adventitious buds in there that trigger all the sprouting. And so then even if, it'd be fine if the stub just sort of stayed how it was and didn't send new growth off of it. But if it's wanting to sprout, then that's another reason to cut it back further.
Gotcha.
Yeah.
All right. Thank you.
My guess is that those stubs are probably dead now. I mean, so they're not probably going to sprout anything. And it is providing kind of that food base for a decay fungus to get in there. So, and you'll be able to tell probably pretty easily where that live collar tissue is now that it's been a few years.
Yeah.
So,
yeah, you can tell. Okay.
Yeah.
Very good. Thank you.
Mm-hmm.
Yeah, you bet.
If you could tell where that live collar's at, could you cut back to it and would it heal over then? This is Philip again.
Well, yeah. Trees do not heal; they only seal over that damage. But yes, if you can, 5-6 years later, cut back to that collar, the tree could have a response pretty similar to it being pruned correctly right off the bat.
Mm-hmm.
Okay, thank you.
You could start correcting it.
Yes.
Yeah.
Did you hear him say they don't heal, but they.
Good.
Yeah, somebody
Thank you.
got cut
Good,
off there. I
good
think.
training.
You bet, Dean. Thanks.
Yep, thanks.
I think we got everybody's info in the chat, so I'll go ahead and close the Zoom, and we'll see you guys either, I don't know if anyone's joining us this evening, but we'll see you Thursday.
Yeah. I'll see you later, Graham.
You bet.
All right.
Thanks, Lori. Good job.
Thank you. Bye-bye.